Hello, my name is Mitchell Brown. Today we are looking at our Unit 2 case studies. The two studies I chose to look at were Case 2 and Case 3. So we'll begin by looking at Case 2. That specifically looks at regulation of glucose uptake by our cells. This case starts off by presenting us with an MRI scan and a PET scan. In the MRI scan, we are shown a glioblastoma tumor that appears in this bright white area. The patient is then treated or administered a fluorescently labeled D glucose. This appears on the PET scan in the bright orange area to show that the glioblastoma tumor has an increased amount of the fluorescently labeled D glucose. So to understand this, we're going to look more specifically at how glucose uptake is regulated in normal tissues. And we will look at 4 different tissue types, being liver, skeletal muscle, brain, and adipose. So we will first begin with liver. So in the liver, we know that glucose is uptaken through GLUT2 transporters. This occurs in an insulin-independent manner and does not have to have insulin present to have the transporter move to the cell membrane. This occurs mostly still in the fed state when glucose levels are high. Glucose will be transported into the liver, used for glycolysis, and also will be stored as glycogen. In the fasted state, we will have glucagon present, and it will cause glycogenolysis to occur of stored glycogen. This will help us to maintain glucose homeostasis in the blood. Glucagon will also cause gluconeogenesis to occur to further the glucose production that is released into the blood to again help with glucose homeostasis. In the liver, free fatty acids are uptaken that will be oxidized as a self-fuel source. This will give the liver the ability to continue to produce ATP to perform its functions. Next, we will look at skeletal muscle. Skeletal muscle uptakes glucose through GLUT4 transporters. GLUT4 transporters are insulin dependent and must have insulin present to bind the transporter to then allow them to transport glucose. So this happens in the fed state when glucose is high and when insulin is high. Glucose will be again used for glycolysis in the skeletal muscles and is also stored as glycogen. In the fasted state, glucagon that is released will have no effect on skeletal muscles because skeletal muscles have no glucagon receptors. But like in the liver, free fatty acids are used to form ATP in the fasted state to continue with cell processes. Next, we'll look at the brain. In the brain, glucose is uptaken through GLUT1 and GLUT3 transporters. This occurs in an insulin-independent manner so that the brain can always be intaking glucose to perform its functions. This glucose is used only for glycolysis in the brain and is not used for glycogen synthesis. The brain, though, still needs glucose in the fasted state, and as it does not get it from these glycogen stores, it gets it from the liver as the liver releases glucose into the blood during the release of glucagon. Next, we will look at adipose tissue. Adipose uptakes glucose through GLUT4 transporters, and again, GLUT4 transporters are insulin dependent. And so this uptake only occurs during the fed state when glucose levels are high and insulin levels are high. This glucose that is uptaken into the adipose is used for glycolysis, but more importantly, the adipose will store this glucose using fatty acid synthesis and tag synthesis. Both of these will result in glucose being stored as fatty acids. In the fasted state, glucagon then that is released will trigger lipolysis to occur of these fatty acids. Lipolysis will cause the release of free fatty acids into the blood. These free fatty acids will be used in the liver and skeletal muscles to form ATP in the fasted state, as we talked about above. So looking at this tumor in comparison to these different types of tissue, we notice that the tumor will uptake glucose in much higher quantities than the surrounding tissues, which is probably true of also the other tissues such as the liver, skeletal muscle, and adipose. As we see in the PET scan, this area of the tumor has a much more dense concentration of the fluorescently labeled D-glucose. So there are modifications that could occur to this tumor to cause this. The main modification that I thought of was the concentration of glut transporters that were present on the cell membrane of the tumor. These glut transporters must be in a higher concentration to allow for glucose to be transported more rapidly. So when blood glucose levels are high, such as in the fed state, glucose levels increase more rapidly in the tumor cells than in the surrounding cells. So with these modifications, the tumor cells have a higher energy store, and this will cause a more rapid growth of the tumor cells than in the normal brain cells around it. And this will lead to a cancerous tumor growth, as we have seen.